too long. Do you want to say something, or?
Oh, just that Steve is very well known throughout the world for his work in functional data analysis and other areas. And the SAMC program you mentioned was a big success, and I think we kind of had things categorized into Euclidean, mildly non-Euclidean, and strongly non-Euclidean data. It was one of the things that motivated me to get interested in networks, which is another reason we're all here around the same semicircle. And I guess without further ado, we should get out of the way.
Well, thanks very much for the kind introduction, and it's great to be back at the Newton Institute. I really enjoyed the program. I've learned a lot about networks. I had no idea what was going on, especially all the probability theory. I thought was brilliant to integrate that. I've also been very happy to come back and make contact with my friends in the stats lab again, so it's been a great visit. Okay, so first, really big picture. As pointed out, if somebody asks me, what are you working on? I'll tell them object-oriented data analysis. Most people will say, what's that? So my cocktail party introduction is I ask people to think about the atom of a statistical analysis. So I teach first courses. Atoms are numbers. We try to get students to think about the variation in a set of numbers. More advanced students, excuse me, will take multivariate analysis where the atoms are vectors. We think about populations of vectors. Very fashionable these days is functional data analysis, where the atoms of the analysis are curves. More generally, I think about data objects. So let me give some examples. For me, functional data analysis is just a special case of object-oriented data analysis, where the objects are curves. A seemingly similar idea is chemical spectra. However, chemists really want those to be non-negative. Most functional data analytical methods require negativity. And so it turns out to be surprisingly different. Anybody who's interested in this, let's have a conversation online, because it's one of my interests. I do a lot of image analysis. You could think of images as data objects. In modern medical image analysis, typically a shape within the image is the data object of interest. I care a lot about trees, meant in the graph theoretical sense, or networks. So I guess this is where I intersect with this Newton Institute program. For me, I'm interested in cases where data objects are trees. I'll show an example in a moment. More recently, I've been getting engaged in neuroscience, functional MRI. I'll say more about that in this talk. Something I haven't done personally, but I really like, is sounds as data objects, which the John Aston group has been pursuing in some very interesting ways. Okay, so let's just look a little more specifically. Functional data analysis. Here's some of my favorite examples. I'm going to look at the one on the right in a little bit more detail. Images as data objects. Here's some of the images I work with. One challenge with images as data objects is it's typically a high-dimensional representation that's required, however you choose to do it, but typically the sample size is fairly low. So I thought a lot about high-dimension, low sample size statistical analysis. Shapes. Shapes typically lie in a curved manifold. A lot of fun there. I'm not going to do much about that today. But if you're interested in manifold data, let's talk about the backwards PCA. Again, I request that you do this offline. Don't have time today. Okay, so what about tree structure data objects? Well, I've been motivated by this wonderful data set where every data point is a set of arteries in one person's brain, and just for fun, I'm a data point, so there's the arteries in my own brain. But we have 100 of them, and so I've thought a lot over the years about how to do statistical analysis of this particular data set. Again, if you're interested, let's talk offline. But I will tell you, I've tried to publish actually 4 different approaches to this, and the one that's worked the best has been topological data analysis persistent homology. There's a lot of skepticism about this in the statistical community, but actually for me it's worked the best of the methods I've tried. So again, if you're interested, let's chat. Okay, coming back to the big picture, what about this terminology? Yeah, I've noticed statisticians are very slow to name things. My colleagues in computer science love names. They name things all the time. I think there should be some intermediate balance, but I've definitely had to defend this name. So here goes my defense of the name. My original idea was I'm trained as a mathematician, so I tend to look for generalities in things I'm working with. So my original idea was object-oriented data analysis was just a mathematical framework sort of containing all these things that I've been working with. But as I got more deeply involved, especially more deeply involved in interdisciplinary work, I realized there was a a deeper value to the concept, and that is it provides a useful focal point for the type of interdisciplinary conversations I engage in a lot with domain scientists. Often I'll talk to somebody who has quite a complicated data set. It's not clear how to get a handle on what should be done statistically, and I found a very useful question is to ask the person, well, of course, I first have to say what a data object is, but after that, I say, well, what should be the data objects here? And I find this often generates the needed type of discussion. Okay, so that's background. Now what about today's talk? Well, today's talk is about joint and individual variation explained. From here on, I'm going to use the acronym JIVE. Somebody said, oh, are you going to give a funk talk? Well, no, not funk, this is JIVE. Okay, so most of JIVE has been developed in a couple of PhD dissertations. First student to work on this was Eric Locke, now at the University of Minnesota. Andrew Noble was a joint supervisor of that dissertation. I'm going to report on the second generation. We've gotten a lot more sophisticated in several ways. The main student doing the work is Ching Feng. She now works for Uber, and she was jointly supervised by Jan Honig. Okay, what's the context? Well, the context is data integration, big data. Oh, that sounds good, doesn't it? Big data. What's big data about? Well, there's lots of different ways to think about big data. My collaborators have big data that comes from combining smaller data sets. And so data integration is a fundamental issue for the type of big data I look at. Now, there's other people who look at online whatever, where they're just big, 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 whatever. That's not where I come from. My collaborators like to put together smaller data sets in several ways. The way of putting together I'm going to talk about today is going to be multiple measurements made on the same cases. One of my big examples, cancer research. I've been doing this for quite a while. 10 years ago, cancer research was all about gene expression. Makes sense, it's a genetic disease. Well, what's going on turned out to be fairly complicated. Just gene expression didn't manage to solve all problems. So what they're doing now is making lots of other different types of measurements, and they're doing it on common people. So putting these together is going to be the focus of what I'll say today. Another example I'm going to look at today comes from neuroscience. Again, how does the brain work? People trying to study this in a data-oriented way. Functional magnetic resonance imaging, measure brain activity is interesting, but now they're trying to connect that up with other things like behavioral feature, genetics, and so on. Again, multi-block measurements made on the same subjects. That's where we're coming from. Okay, so let's just be clear about where I'm coming from. There's an organizational model here. First, for a moment, just think about a single data set. Natural often to form that into a matrix. I realize networks is about not taking a matrix approach, but let's consider cases where you can organize data into a matrix. And now here, let's be careful about what's going on. The convention in this talk is I'm going to use columns as data objects. So think about those as columns. These are going to be the atoms of the analysis or the experimental units. Now, if you do a lot of work with, say, SAS or R, you're used to the transpose. You're used to rows as data objects. I like columns because I'm mathematically trained. I want my vectors to be columns. That's the only reason, but I will say most statisticians are used to thinking about the transpose here, so you're going to have to be careful, but it's my talk, so I get to say how we're going to do this.
Let's turn the slides.
Yeah, we'll see who turns their head as they listen to the talk, yeah. But you know, I find quite often when I get into a conversation with somebody, once you get into the linear algebra, so often we're talking away and then I realize, wait a minute, I'm thinking columns and he's thinking rows. And so I often try to be right up front by saying, what are the data objects here? Are they rows or are they columns? Okay, so notation, I'm going to take D to be the number of variables. A lot of statisticians like P. If you want to argue about that, we can, but I'm going to use D. That's exactly why I'm doing this, getting you thinking here. Okay, so N is going to be the number of cases. Okay, now, one key assumption is I'm going to assume mean centering has been done. Again, think of vectors as objects. I want to subtract the mean in such a way that if I take the mean of the columns, I get the zero vector. Okay, so mean centering. Now here's where it gets slippery. You could, if you like, think about that element-wise, and that operation of taking the vector mean, you could think element by element, take and subtracting the mean of each row. But again, objects are columns here. Okay, now with JIVE, what we want to do is the same kind of thing, but I want to start looking at multiple guys here. Data types, or I'll often call those blocks. Okay, and the important part is measurements are made on, say, the same people, so the columns are the data objects here. Okay, so first let's just consider the only 2 blocks. I'm going to do multi-block a little bit later, but some of the ideas are best introduced with 2 blocks. So I'll focus on that for a while. Notation, I'm going to have D1 and D2 as numbers of features. Of course, they can be different. In fact, you'll see often they want to be wildly different. Okay, so there's the notation. Now, what is it I'm going to do? Well, I'm going to now explore and quantify variation in the sense of principal component analysis. Now, I've come to realize when I say PCA to most people, they think, oh, dimension reduction, he's just going to reduce the dimension. Well, actually, there's a lot more going on to PCA than that, which I'm going to illustrate with a particular example here. Give you a feeling for what I mean by explore, quantify variation in the spirit of PCA. So just to get you thinking in that direction, I'm going to look at a single data set, fun example called the Spanish male mortality data, and I got this from Andres Alonso at University of Carlos III in Madrid. Okay, so here the data objects are just going to be vectors. What they're going to be is mortality. Mortality is your chance of dying as a function of age. So age is coming down, and this is just demographics. Just search through the database of who died. Look at the age of people who died of a given, or look at the number of people who died of a particular age, divide that by the total number of people of that age, you got the mortality. Now this varies over several orders of magnitude, so I'll take the log. Notice choices of data objects. Look on the log scale. Okay, then for fun, we have this for a number of years. I'm going to look over basically the 20th century, essentially. And we'll have fun understanding how mortality changed over time, and there's an interesting history. Okay, so the first way people look at functional data, curves as data, is they'll just look at an overlay of the curves. So there's an overlay, and you can already see some interesting things. First off, I think I'm going to skip here. Yeah, first off, it's dangerous to be born. Infant mortality is a serious issue. If you can get past birth, your chance of dying goes way down, at least to about age 10 or so. Teenagers, well, all of a sudden you find a bit more risky behavior, it kind of goes up. Maybe the brain myelinates and it levels off. And then, of course, well, people get older and their chances of dying definitely goes up. So you can see all those things there. So that's fun. But what you can't see is the change over time. Hard to see time structure. I'm going to tease out time structure just by changing the colors. I'm going to color these things in time order using a rainbow color scheme. So I go from purple at the beginning through blue, cyan, green, yellow, red. And now you see what's pretty obvious. Things have gotten a lot better in terms of mortality over the last 100 years. People's mortality has really fallen off. Most of that's due to improvements in medicine, public health, stuff like that. Okay, just for fun, I'll color code the years there, just so you get a feeling. So in principle, if your eye is good enough at distinguishing color shades, you can actually go back and identify each of those. Okay, Patrick.
Maybe you're going to get on to talk about this, but I was curious about how much the sample size changes as you go through, and maybe you're going to talk about variation as a function of age, or
I'm certainly going to talk about variation as a function of age. I'm not going to talk about sample sizes. I'm just looking at ratios. And so notice we cut this off at about 95. The data goes up into the hundreds, but then you get into data sparsity issues, zero denominators, and all that stuff. So this is enough so the ratio is really where the action is. Peter?
What about variation as a function of time in the century? For
Yeah.
instance, the Spanish Civil War or something like that.
Absolutely. You're giving away some of my stories at the end here. I'll totally come to that. Now, you're also asking an interesting question, which I'm not going to talk about, which is, what if your variation is about time shifts? That's an important mode of functional data analysis I'm not going to talk about. If you're interested, again, it's something we can talk about offline. But this is a data where that's not a particularly useful way to look. In other data sets, it becomes crucial. Richard?
So one thing that's quite interesting about this, of course, from, say, roughly age 30 to 90, you've got this log linear increase in the hazard function, right? So that actually corresponds to the left tail of a Gumbel distribution in terms of the underlying distribution.
Wow.
Which is like the asymptotic distribution of the minimum of a bunch of normal random variables. So it's like, you can think of it like you live your life and you've got these swords of Damocles that hang over you.
Yeah.
And at a certain point, one
of them gets you, yeah.
and you die.
Yeah, yeah. You know, I work with people in prostate cancer, and they say prostate cancer is a disease every male will die of unless something else kills him first. But they stopped saying that after it was pointed out, well, isn't that true about every cause of death? And I guess that's exactly your minimum.
But so actually, I mean, this left tail of the Gumbel distribution, incredibly light tail, it's iterated, this double exponential tail. It's
Interesting.
But typically, if you go beyond 90, actually it tends to come back down. Otherwise, you'd never see anyone over 100.
Yeah, right.
If you survive up to a certain point, then
Wow. Really interesting considerations I never thought about before. I'm going to look at different aspects, but thanks. I really appreciate that comment. If you like, we could take a look at bigger. I've got the data for it. So, wow. Okay, fun. All right, so now, as a statistician, the way I think about, oops, let's go back. The way I think about this set of curves, I find it useful to think about how they relate to each other. So I like to think of each of these as a point in a point cloud, and then think about the variation in the point cloud. So what's the dimension? Well, it's about a 95-dimensional point cloud I'm going to think about. Okay, so I got this point cloud of data. First thing a statistician to think about is what's the mean or the center point. So that's easy. Just take the mean, show it to you as a curve. So notice you can see some interesting things in there, such as dangerous to be born, gets a lot better, risky behavior, all that kind of stuff. Richard's Gumbel-generated curve, all that stuff is there. So in fact, all of those things appear in the mean. That's what you can see at the beginning. Oops. Now, what about those blips? Well, at first you might think they're noise, but look more carefully, they're periodic. Not only are they periodic, in fact, they appear at decades. And so what's going on here is, this is why Spanish data are so interesting, and things like German or Swiss data are pretty boring. It's all about record keeping. In fact, in the 150 years ago, they didn't keep very good birth records in Spain. So if somebody died, they say, he was about 50. And so you can see that 49 and 51 are low, and 50 is a bit big, just because of that age rounding effect. And you can even see a little bit at the 5s as well. Okay, so that was fun. Now I got my point cloud. We found the center point. Let's study variation about the mean. So I'm going to take that point cloud and shift it back, look at the curves that you get from that, and you get those interesting curves. So notice, first off, most of those age effects are not here. So in fact, that's not variation about the mean, it's in the mean. That thing is there for all the curves, and it's about the same. But of course, what's changed is things have gotten much better over time, and furthermore, better for young people than it is for old people. Medicine works great for the younger people. There's a law of decreasing returns for older people, I'm afraid. Okay, so main effector in the mean, not the variation about the mean. Okay, now I want to kind of focus this, because when you look at that, there's a lot of stuff going on there. You understand the main effects, but let's try to focus the analysis. What I'm going to do is take this point cloud, and I'm going to project on the direction of maximal variation. That's the first principal component. So if I project everything in that direction, if I look at those projected curves, I get a family of curves. That's what you're looking at here. There's the PC1 projections. So notice the colors correspond to the different coefficients, but they're all multiples of the same curve. Peter?
I didn't quite understand. You said there's effects of age in the mean, but there's no age effects of variation around the mean?
Oh, there's an age effect. Sorry. What I meant was those features of dangerous to be born, and then mid-childhood is the best, it gets worse. All of that kind of structure is only in the mean, not in variation about the mean. There's still age effects, of course, because there's more variation for the young than for the old.
I was just trying to reconcile the picture with that.
Okay, good. Thanks for clarifying. Okay, good. So now we've got we got this. Okay, so notice we've certainly captured the major mode of variation there. That's all good. And that is pretty much everybody's up, everybody's down. Of course, that's more true for the younger people than for the older people. But we see that's the main mode of variation. Okay, because I'm going to use this later in the plot, I'm going to give this a name. This I'm going to call a loadings plot. In principal component analysis, the loadings are the entries of the eigenvector. So what I've done is each of these curves follows the loadings of the eigenvector multiplied by the projection coefficient. So they're all multiples of the same curve. But keep in mind, we got this point cloud in that direction, because that's going to be become important in a moment. Okay, so medical technology, everything got better. Notice the age rounding blips go up and down. This is because record keeping improved. At the beginning, there were age rounding effects. They kept better records, it went away. Keep in mind, I've subtracted the mean. So the contrast here shows an up component early and a down component later. Okay, now what's going on with those different curves? Now let's start thinking about the colors. It's mostly a monotone color change, but in fact, you can see some other stuff going on. So I'm going to focus on that now, and I'm going to do it by looking now at the scores. So the scores are just the coefficients of the projection, and I plot these on the horizontal axis here. And then what's on the vertical axis? Well, two things are going on. First off, I just plot, I'm just coding for order in the data set. So increasing height just means increasing time. You can see the colors correspond there nicely. And then, okay, so let's look at that for a moment. Okay, so look at the dots there. So the first thing you can see is overall improvement. The rainbow's going off to the left there. Things have gotten better over time. Higher mortality earlier, lower mortality later. That's great. Now what about that black curve? Well, the black curve is a smooth histogram or a kernel density estimate. It's just showing that the dots are dense here, they're dense there, and it highlights the fact that it's quite sparse in the middle. So what that means is the relative improvement happened over a fairly narrow time frame, mostly again medicine and public health. Okay, so that comes out nicely. Okay, now you've probably noticed some outliers there. How about that purple one? Anybody have a guess of that? Pardon? Oh, very good. If you study the field of epidemiology, the big event in the history of epidemiology was year 1918, just after World War I. Everybody traveling around a lot, a horrible strain of flu spread around the world. Millions of people died, including Spain. How about some other structure there? Well, somebody already gave that away. You know, I show this in the United States, and people guess World War II. It sort of shows a typical American education, I'm afraid. But of course, Europeans realized it was 1936 to 39. There was a horrible civil war fought in Spain, and that actually is the Spanish Civil War you're looking at. And then things pretty much look better. Okay, so we got our point cloud. We found the direction of maximal variation. There's some good stories. Now let's look in the second principal component direction. Think of this as take the point cloud, project on the orthogonal plane, find the next maximal direction of variation. Okay, then look at the loadings plot. So there's my loadings plot here. Now this direction has to be orthogonal in this space. And so the first one had all the components positive. You're going to have something orthogonal. Some are going to have to be positive, some are negative. And indeed, you're seeing that. Now, how do we interpret it? Well, notice, fortunately, it's just a nice contrast between the 20 to 45-year-olds, and then there's the union of the young and the old. So contrast how how the middle group compares with the union of the old. All right, so you got the idea. Keep that in your mind. Now, what about colors? Well, you can see some patterns in the colors, but it's not so easy. It's nicer, again, to go to the scores. So I'm going to go to scores again, and that'll be more interpretable. And now you can see some interesting stuff going on there. There's the flu pandemic. Well, 20 to 45-year-olds died at an even greater rate relatively speaking. So that's why that one shot off to the right there. Spanish Civil War, you can see, of course, these are guys actually fighting, so of course it was hardest on them. And then you might think, well, once the war was over, things would get a lot better. Well, if you know your Spanish history, you realize that the Franco regime won, and I'm afraid they did some things that were not so nice, like revenge killings and things like that. And so actually it took really quite a while. Now, the revenge killing stuff, that happened more with the 20 to 45-year-old males, and so it took quite a while for that to get a lot better. Fortunately, it did. Okay, now note, it went back the other way. Anybody have a guess about this? Here we're talking 1950s, 60s. There's a very important sociological event that happened gradually during that time period. I guess none of you are old enough to remember when not every family had a car. This is actually the introduction of the family automobile here. And who is it who drives like crazy on wild Spanish country roads? It's the 20 to 45-year-old males. So in fact, increasing automotive death rate. Now take a more careful look. Notice it went back. Why did it go back? Well, now I'm sure there's people in this room who remember cars with no seat belts. We don't have that now, but everybody can, not everybody, if you're old enough, you can remember this. And so in fact, it's increasing car safeties. Cars are much safer than they were, say, 20, 30 years ago. Not only that, the roads are safer as well. So this is, the corner is finally turned by safer cars and roads. What about the future? Oh, Shaheen?
I mean, I guess you can go and, do you have data about like the cause of death?
We don't have it.
Okay.
So, yeah, stratifications like that would be possible. We got this data from a website that just specializes in published mortality.
Okay.
And they put more of their energy into more countries than they have cause of death. So I don't know where there'd be a source of that. It could be interesting to look
I guess you'd see at least something just from having the data for females as well. Right?
Oh, we do have the data for females. And in fact, something I'm not going to have time to show is a JIVE analysis combining males and females. It's actually fun, but I don't think I'm going to get there today. Okay, so what about the future? Well, I wonder, texting and driving is becoming very popular. Texting can be quite dangerous. I wonder if that's going to show up. Don't know the answer yet.
Tesla.
Yeah.
Teslas.
Okay, so now let me put those things together into my usual view of data, which is a scatter plot matrix view. So notice I got the PC1 scores, PC2 scores, one-dimensional views are on the diagonal, corresponding two-dimensional views are off the diagonal. Now these are carefully linked. This axis is that axis. The axis there gets flipped up, so it really is the two-dimensional view. These are one-dimensional views, that's a two-dimensional view. Notice it really shows the structure well. You can see the rainbow pattern coming down, and then you can see those variations about the 20 to 45-year-old. This is just the transpose of that. So no new information. Sometimes one is nicer, sometimes the other. Okay, so that's my common view of data. This is the scores plot. So with PCAs, you got loadings, you got scores. Scores tell you about how the data objects relate. Because there's a time ordering, I've connected with line segments. A lot of data sets, that's not the case, but here it's a useful visual device. Okay, so again, this is my common first graphic when a new set of data comes into my office. And notice what it does is reveals relationships. Okay, second graphic is, when you look at the scores, you often want to know what's driving it. So that's where these loadings plots become interesting, as I showed you earlier. Okay, now let's get to JIVE. So I've given you a feeling of what I want to do. Now I'm going to show you how we're going to do this for two or more data sets. So now you understand what I mean when I say in the spirit of PCA. But we just do it for more data blocks. Okay, so the JIVE part is, what do you mean by joint? Well, by joint, I mean variation of that type where they're working together. And I'll define what I mean by working together in a moment. And furthermore, not only do I care about this kind of variation where they work together, I also care about variation where they're working separately, really independently in a particular sense. And of course, I have to define all this for you. Okay, so let me next do a toy example here. So this is just data we constructed. I'm taking a heat map view here. So it's a data matrix, and I'm just showing you the entries of the matrix with colors there. Red is positive, white is 0. Blue is negative, and the intensity shows magnitude. Okay, but of course I got two of these data sets. So you can see I've got a couple of them here. All right, now where do these come from? We deliberately cooked these to be quite challenging in this domain. Challenging aspects are the dimensions are way different. First data matrix has only 100 features, the next has 10,000. Also, we're doing really different noise levels. So the variance for one is much bigger than the variance of the other. Okay, so wildly different sizes and scales. Well, I don't apologize for this because it's actually typical in cancer and neuroscience data, as I'll show you when I get there. Now, sometimes people say, oh, if you got different scales, it's no problem. Just take and divide each row by the standard deviation. Well, if you have similar numbers of features, that will work great. When you have wildly different numbers of features, it's much more problematical. You make a very specific, make a very specific emphasis choice when you do that, and you may well be emphasizing away what it is that you want to see. You could also work with the Frobenius norm, but again, is that the right normalization? Frobenius norm is just the total sum of the squares over the whole matrix, but again, is that the right normalization? Well, a very important contribution to the methods we developed is we can actually avoid having to think about that in a sense that I'm going to show you. Shaheen, you look like you have a
Just a
question.
question. So the noise is different, but the signal to noise ratio, is it about the same in both matrices? So I mean, do you have like the entries, the uncorrupted entries, they are the same magnitude or no? They are proportional in magnitude to the
Well,
variable?
there is a question of how you even define signal to noise, but it's a red herring. It's not important to what we're going to say for reasons I'm going to show you. So I did not make a deliberate attempt to do that. I guess there is a sense in which it probably is happening, but that's not going to be important. Okay, so where'd all this come from? Well, the underlying components are just a simple additive model there. We've got a clear noise matrix. That's just IID Gaussian noise. Projector doesn't show that so well. We'll see what that means when I get into more complicated stuff. But how did I choose those? Well, first off, the joint component was chosen to be just a rank 1 signal. Oh boy, you can't see that very well. All right, so what we're seeing is blue and red. And so this is a rank 1 signal matrix. Oh, that's better.
I guess you can close that door.
All that down.
It's okay. I think we can do it.
We're getting there.
That's good, yeah. Don't get too dark. I can't see who's asking questions.
Is that
Okay,
that's
I can still
actually
see
a bit better,
people.
right?
Yeah, that's better, yeah. Okay, so what we're seeing is blue and red. It's a rank 1 matrix. The left vector here is mostly zeros with a few ones here. The right vector is plus ones and minus ones, so that's rank 1. That one has a different left vector. The right vector is the opposite. It's red and blue. And so the point is that it's just pluses versus minuses, minuses versus pluses. Those are the same subspace. And so that's the key. That's the sense in which this is joint. And now, oh boy, this gets a lot worse. Okay, I'm going to have to tell you what's going on here. I have 4 blocks of alternating colors there, which you can't see very well. This is rank 2. I've got 5 blocks of different colors. And here I've got 4 colors again of different colors. Okay, I'm going to have to go quickly through this because you can't see anything. So there's an individual X matrix, which is rank 1. That I've took to be orthogonal to the joint. And now here's the hard part. It took us a while to understand this, but the right notion of orthogonality is orthogonality in the sample space. So it should be in Rn, they should be orthogonal. And unfortunately, can't see that very well.
Do you want some? Is that your?
That's my water,
okay,
yeah.
Just checking.
Yeah. Okay, so it's orthogonality in the sample space. Okay, great. The individual matrix is rank 2. Again, you can't see that very well. Should be orthogonal to the joint. And again, it's in Rn that the orthogonality happens. Then we have to have the relationship between the X and the Y. At one point, we thought it had to be orthogonal. We realized that didn't make sense. In fact, all you need is the intersection not to be 0, but they don't have to be orthogonal. So that that's an important aspect of that. Okay, now let's try some. Well, I'm going to skip through this pretty quick, I guess, because you can't see anything. One way to analyze these data is say, well, why? Who cares about this? We'll just put them all together, combine all the data, concatenate, and then just do, say, PCA or SVD. You see anything there? Not much. A lot better shows up here, but anyway, what you can see is that the rank 2 is a pretty nice denoising of that in the X space. So in the X space, it works pretty well. Now, I don't show you rank 1 because it's not very intuitive. It doesn't do anything like the joint in the individual. It just does its own thing. But rank 2 looks pretty good. Now, what you really can't see is that what happened in the Y space is a lot more disappointing. What you get is actually feeling the joint signal and the individual signal in the X space. It feels what's going on in the X space, and the reason is these coefficients are so small here. Okay, so it can't really find stuff very well. You come up with a good reconstruction of the joint in the X space, but not in the Y space. You lose the separation, and the Y individual just doesn't work at all. Okay, another methodology is partial least squares. Well, I think I'm just going to skip through this stuff because you can't see anything. But I will tell you partial least squares also has serious problems. Partial least squares attempts to find directions in the X space, directions in the Y space with maximal covariance. In a lot of situations that works quite well. We've cooked this because we have to show we're better. And so we've cooked this so it doesn't work that well. You can't really see it, so I'm going to skip this. Early JIVE. Well, we want to publish a new paper on JIVE. We have to show that it's better than the old one. And so we did that. And basically what happens is if you use the early version of JIVE, you get a very good rank 2 or rank 3 approximation. But where it tends to fail is it doesn't get the individual and the joint quite separated correctly. So that's another important improvement that we've made. And again, the early version depended on normalization, which we don't need anymore. Okay, so good 2D approximation, but we missed the joint and individual. So here's the current version. Oh my gosh. I will tell you, this looks really good, and we'll just leave it there. So we get a great decomposition. Okay, well, so much for the background. Now that I've shown you that I have a method, let's try it out with some real data. I'm going to take a look at some functional magnetic resonance imaging data. So here the idea is measure brain activity over both location and time via blood flow. I like this for object-oriented data analysis because there's lots of questions of what should be the data objects. So my collaborators are Chun Yu, my PhD student, and Ben Risk, who's a SAMC postdoc. Okay, so the main idea, think about taking locations in the brain, measuring activity over time. And so it's, in some sense, a four-dimensional representation. And there's lots of issues about data objects. Do you want a 3D movie? Do you want to focus on individual voxels and look at the time series? Lots of stuff that can be done there. Okay, so we're going to look at data from the Human Connectome project. These are a large group of people who've, I guess, spent a lot of money coming up with some quite useful data summaries. There's a lot of work to be done there, and we're just totally leaning on them for that. So the data set we're going to look at, there's 491 people. That's fairly ambitious. We're going to look at behavioral features. So these are things like cognitive scores and so on. We're just going to look at 139 of those. And the image data we're going to look at is going to be three-dimensional voxel summaries for the task of working memory. And so what they've done is taken the three-dimensional image of the brain, and they've done a whole lot of work registering these across people, making the volumes correspond across people. And then there's lots of voxels where not much activity is happening. The activity is measured in terms of blood flow. And so a lot of the brain is just fluid. There's no action there. So we're going to ignore all of those. They've done a lot of work to come up with a set of voxels that are thought to correspond pretty much to the folded cortical surface of the brain, and we got about 90,000 of those. So it's a lot of data reduction that's been going on, but still we end up with fairly high dimensional, which makes sense. Okay, so again, there's registration that happened. All those problems have hopefully been solved in a reasonable way. Okay, now we're going to look at behavioral data. So here I take the behavioral data and just look at the separate PCs. So I'm taking my point cloud of behavioral data, finding the direction of maximal variation. And well, there's a lot of stuff there, and I have more time, I go into more detail. But you can see groups of features that are colored. Yeah, so there's important groups there, and if we have more time, I go into the intuition of that, but I think I'll skip that for now. And notice what I'm doing is I've got my point cloud of behavioral score, I'm taking that direction vector, and I'm just showing you the entries of the vector. So what that says is that people in this sort of aqua group, when those features go up, the others go down, and this makes intuitive sense, but I won't go into it. Okay, now I'm going to do the same with the image data. So we've got set of image data. And so now what I'm showing you is the loadings plot of the first principal component of the image data. Okay, so again, this is just the entries of the direction vector, and the visual display I'm looking at goes like this. The human brain has these two halves. If you cut it in the middle, the connective component is that small thing, which is the gray area there. And then each hemisphere, we're looking at one side, looking at the other side. So 2 hemispheres times the 2 sides, you get the 4 things there. Okay, so we can see some stuff. And then all I'm doing is using a color coding here for the loadings. Notice most of them are hot colors. Very few are cool colors. So what that says is this mode of variation is working in some sense like what we saw with the Spanish mortality data. They all go up and down together. Okay, so that's the entries of the eigenvectors, and the first thing I saw with that is, hey, they're all going up and down together. That's not unusual because some people are just going to have more, well, I better not say more brain activity. Some people are going to have more blood flow relatively than other people. So it's just an overall amount of blood flow. You could think activity if you like. Okay, also there's some hot spots there. Now, what about those hot spots? Keep those in mind because they're going to become important. Now let's look at the second component. So I took my first eigenvector, I look at its direction. Now I take the second eigenvector, look at this direction. It's going to be orthogonal. So now we're seeing both hot and cool colors. That's good. Did you keep those hot spots in your mind? Take a look at those hot spots. Let me go back to PC1. Notice it's the same hot spots. So what that says is we got a confounding here of the direction representing the hot spots with the direction representing up an overall variation. So that's not a nice decomposition. You could think about doing maybe a Varimax or something to try to separate it, but I'm going to take a different view. Okay, so keep those hot spots in your mind. Now the question is, what are they? Well, there's been previous work done, and I'm going to show you a map constructed from previous work. And in the previous work, I'm showing regions that are associated with working memory. So now let's come back. Notice it's pretty much the same hotspots there. Okay, so this is good. We seem to be finding something important. Now, what about the jive? Let's do some jive. So what I'm going to do is a similar analysis, but I'm not just looking for directions of variation. I'm looking for directions of variation that are associated with behavior scores in the jive sense. So that's going to be the next step. And there's the jive PC1. Okay, so notice it's the same hotspots. And as I jump back and forth to the given map, notice it's really very similar regions. So we've done a good job of disassociating the confounding of the two directions there. Okay, now let's take a bigger picture point of view. There's lots of different analyses going on. So I'm going to sort of lay them all out here like this. All right, so what I've shown you before, we started with the PC1 of the, I had a stick here now. We started with the PC1 of the behavioral scores. That was this one. Then we looked at the PC1 for the images. That was this. We looked at PC2 for the images, and then I looked at the joint. So I'm laying all this stuff out here. First is just separate analysis, ignoring how the blocks interact. We'll look at the joint part, and we'll look at the individual part. So there's all of those things laid out there. Now there's a lot going on. So let's take a look. Interesting features are, take a look at the PC1s. The eigenvector for the separate PC1 looks a whole lot like the joint. This says they did a great job of choosing the features. We're not looking at all the behavioral scores. We're looking at behavioral scores that are thought to be associated with working memory. And so notice the variation that's natural just in the scores is joint. It works together with the joint variation. So that's an interesting thing. Next off, take a look at how the PC directions come in. I said, well, we had both overall variation and hotspots, similar hotspots. Notice the joint is using the correlation with a separate direction to come up with an improved direction, which ignores overall variation but really focuses on the particular aspects that we care about. And then notice the individual plays a role here too. I suggest it's not enough just to look at joint. You need to pay attention to individual as well. Notice the overall up and down is nicely captured by the individual. So it gave a very nice decoupling of these two directions that were present there. And then we go further, now we get to a second mode of variation. Now we're getting to the point we can tell the neuroscientist something, because this is an orthogonal direction, and it's something they don't know about yet. But notice we have a nice capability to sort those things out. Okay, so now, JIVE algorithm. Oh my gosh, 10 minutes left. I was going to go through a very detailed discussion of this, but I think I won't be able to. I'll give you the overview here. So here's how things work linear algebraically with a JIVE. You know, when we wrote the JIVE paper, we just sort of tried to describe all this. And then Ben Risk goes, oh, if you're going to publish in the neuroscience world, you have to have a path diagram. So we thought about it, and actually we realized the path diagram was really helpful here. Okay, so I'm just going to give you the 10,000-foot summary here. We start with the data matrices. First thing we do is a low-rank singular value decomposition of each one separately. Okay, then when we've got that, we focus. Now recall the linear algebra happens in the row space here, so we focus on the two row matrices. We find the joint components using something called principal angle analysis. This is a useful property from linear algebra, which I'm not going to take the time on. We do principal angle analysis. Careful thresholding, there's a lot of work that went into that. That gives us the two joint spaces. Then we essentially do subspace subtraction to take the full space, subtract the joint to get the individual space, do that for both of them, and then we do a reprojection to come back to the final result. Okay, so now you may want to close your eyes while I go quickly through all these things. Anybody who cares about this, I'd be happy again to go into details offline. Okay, oh, output. Very important thing about the output. We have several different versions. I'm going to skip to here. One possible output is you want only the scores. Now recall there was these normalization issues. If you want to avoid the normalization issues, you work completely just with what we call the common normalized scores. Now for other purposes, you want to take scale properly into account. So you use what we call block-specific scores, corresponds to both of those. And in other situations, you want to get back to the full data matrix. For example, if you want to get back to the original features, like you're doing gene expression, you want to start talking about genes, you need the, you need the full data matrix output, all of that. Okay, so then there's the extension to more than 2 blocks. Here we, okay, I'll just skip that because you can see that anyway. We can do it. Okay, now one more example I want to get to is some cancer genetics. So data here came from the Cancer Genome Atlas. This is a large-scale multi-lab approach to trying to gather large data sets. The problem with cancer, challenge of cancer research is that any given lab can generate typically low hundreds of data. And so what you can do with that has pretty much been done. There's been a big effort to try to get labs to work together. One of these is called the Cancer Genome Atlas. And so you can see they're very ambitious about a dozen different cancer types, and they're making lots and lots of different measurements. Networks. Notice cancer people love to think about how to organize these things in terms of networks, and that's going to be the one of the open problems I'm going to get to in a moment. Okay, so let's take a look at breast cancer data. A lot of work going on, a lot of intersection has been done. While the data types are supposedly measured on everybody, there's missing data and stuff like that. So doing appropriate intersections, careful cleaning, we came up with a data set with 616 people, and we're going to now study 4 different types of data. So it's going to be a multi-block JIVE that I'm going to do here. All right, now an interesting feature of breast cancer data, people have been thinking about this for a long time, is there are important tumor subtypes. These tumor subtypes have a real impact on treatment. Classical treatment for breast cancer is carbotaxol chemotherapy. It can be very effective for some people, but it's known that there's a set of people who go through that grueling treatment and don't get any benefit from it. So my collaborator Chuck Peru is famous for discovering the luminal A subtype. Luminal A's are the people who not only get no benefit, they have the best survival. So they're the people you do not want to give the carbotaxol chemotherapy to, and there now exist tests on the market for that. But anyway, these subtypes enable me to keep track of how we're doing with the JIVE analysis. Okay, so first let's do a separate PCA on the gene expression. Notice this very nicely separates out the groups. The basal appears on the right in PC1. In PC2, we do a good job of separating the luminal A and B. So that all looks really good. Well, actually, that's not very surprising. You can see clear subtype effects. That's not surprising because the subtypes are actually defined from clustering such data. Not this data set, but related data. Okay, so PC1 flags basal, PC2 is the luminal A's. Now let's go to a different data type. So here's a separate PCA for copy number. This has the subtypes, but not so strongly. Now they appear mostly in the third principal component. Okay, so again, there's subtypes. All right, let's do some jive. We've got to do some thresholding. I'll skip that stuff. Okay, I think I'm just going to skip this stuff. There was some nice stuff that happened. Here's what we got from the four-way joint analysis. We ended up with the luminal A's on the right. Notice if you just do PCA, it's the basals that pop up. What this is telling us is that the difference of the basals from the rest does not appear in terms of all four of these measurements. If we're going to do all four of the measurements, in fact, what pops up is the luminal A is more important. Okay, so that flags the luminal A's, driven by all four factors together. And so actually this is about mutation. In fact, what we've shown here is that the luminal A's are about mutation, and that difference of the basals does not appear in the mutation data. Okay, so then we said, well, all right, let's, mutation is not that interesting. There's pretty much one component. Let's get rid of that and do a three-way jive on the remaining three blocks, the gene expression, copy number, and proteins. Now here's a place where I think we have some room for improvement. We did this just by taking the individual components that we got from the four-way jive analysis. So that's our way of getting the mutation out of there, as opposed to just throwing it all away. We want to look at what's kind of orthogonal to the mutation. So we used the three-way individuals from above. We did jive to the joint PC1. Now notice that the basals show up. And furthermore, we're getting stronger distinctions between the groups than what we got before. Okay, so you can see the HER2s in the middle. A's and B's are combined, but that's okay. Okay, so notice the separations we're getting looks a little more distinct. Well, okay, that's visual. Can't we get a bit quantitative about this? Can we really measure this? You can sort of see it, say, oh yeah, it looks kind of different. Or you could be really a skeptic and say you're not sure. But let's think about getting a little quantitative about that. Okay, so now I'm going to do a different analysis. I'm going to do a pairwise jive just on gene expression and copy number only. And to get quantitative about how this method's working, I'm going to construct some, well, essentially p-values here. I'm going to use diproperm z-scores to give me a quantitation. Okay, so now I look at the same gene expression as before. I'm just going to study how the basals are different from the others. So I showed this picture before with the basals in blue for copy number. Recall the action was in the third component. And then I'm going to take all the union of the other cancer types and just color it gray. And I'm going to study blue versus gray. Okay, so here's a rotation of the data. Here I did a PCA. This is just separate copy number PCA. Often if I can see some difference, I'll say, well, you know, this is just PCA. Principal components feels variation. It doesn't feel class labels at all. So what I'm going to do is introduce a new direction, which does a good job of separating the data called the DWD direction. So DWD separates the data quite well. Notice the separation you're seeing here is better than what we saw in the others. And then I'm just using orthogonal principal components. DWD, look in the orthogonal direction, take the principal components in that direction so that they're all orthogonal. If I don't, they're not orthogonal, it doesn't look so good. Okay, so now, how are they separated? Well, visually looks pretty good. Let's try to measure it. I'm going to measure it with something I call the diproperm hypothesis test. Quite often, when working with high-dimensional data, if you use something like DWD, it'll often really separate things, but that separation can be spurious. It's important to do some inference. The inference I like, I call diproperm. The idea is, what do we got? Well, we took the high-dimensional data set, we found a direction, we did the one-dimensional projection, we saw that separation. How important is that? We'll assess that with a permutation test. So what I'll do is first measure how far the two clouds are separated. One way to do that is the difference of the means. In this case, that's about 28. Now, what does 28 mean relatively? Well, I take that number 28, put it as a green line over here, and I'm going to take the data and randomly permute. So I'm just going to reshuffle the labels, recompute the direction, reproject, recompute the mean, and I get the black dots. So now what you're seeing is there's a null distribution of black dots way down here, around 2 to 6. I came up with a number like 28, so that's strongly significant. All right, so that's okay. Now what I want to do is I want to compare these across settings. Well, a challenge with a comparison across settings is I'd like to use the p-value, but notice that the empirical p-value is 0. I'd have to do a whole lot of simulation to get anything to pop up beyond the green line and give me a p-value I can use for comparison. So instead of using the p-value, I'm going to use the z-score. Just take the black dots, look at their mean, look at their standard deviation, ask how many standard deviations above the mean am I? In this case, the answer is 27. So I'm going to quantify separation with the z-score, and that's what I'm going to use for my comparison. Okay, so that number 27 is just the height of the bar there. And so here I'm showing you separate analyses. If I do diproperm on copy number, I get 27. If I do it on gene expression, I get a much bigger number. Well, that's not surprising. The gene expression data clearly showed a much better separation, so that's good. Now, what about JIVE? So now let's do some JIVE. Let's understand how gene expression and copy number are working together. Well, the nice news is, that's not surprising. The nice news is, if I incorporate copy number information using JIVE, then I repeat the analysis, I get a stronger difference. The z-score goes from 69 to 78. Okay, so this is the effect of combining the information. Notice copy number also gets a boost if you combine that. Now what about the individual components? Well, the gene expression, surely a lot less. So what that says is that the discriminatory information in the gene expression, it's smaller if you subtract out all of the copy number part, but it still gives a z-score of about 9. So it's still statistically significant, which is not surprising. It's expected there's stuff going on in the genes which is not going on in copy number. And finally, if we look at copy number, there's nothing at all. So what that says is all the discriminatory information in copy number is shared by the gene expression. I didn't find anything after that. Okay, well, there's a lot more of that stuff I could do, but I think I've given you the main idea. All right, yeah.
So can you draw the conclusion that you can just throw away the copy number information?
Well, not quite. I mean, it did give a boost. So I wouldn't quite throw it away to give some boost. And by the way, this is only one of these comparisons. There's lots of others where, in fact, it actually is beneficial, but I just don't take the time. But yeah, you have to be a little careful how you interpret these things. And I'll be honest, we're still sort of working through it. Okay, so let me just mention some open problems. This is a workshop on network analysis. Here's where I'd love to think about some serious collaborations with networking. So a common tool in a lot of this research is understand how the variables react with each other. You're doing gene expression. How do the genes interact? There have been a number of talks on this type of topic. What I think would be quite interesting would be to think about doing a post-JIVE gene network analysis. In particular, do some networks on the joint and the individual and see who's where. So for example, a standard analysis, let me turn that off. I should be able to do, well, anyway. Okay, so a standard analysis is take a matrix, do some network analysis. You can do that on the separate, everybody does that. I'm quite interested to see what happens if one tries that analysis on the joint and on the independent, and understanding how these networks relate to the different data types is where I'd like to go. Even more sophisticated would be to take and integrate network ideas with the JIVE linear algebra. I think this is possible as well. All right, we've also heard a lot of talks on brain connectivity. So I think it'd be quite interesting to combine JIVE with brain connectivity stuff there. Relationships with behavior and genetics. I think it's a wide open area. If anybody wants to talk further, let's definitely do that. JIVE specific stuff, there's more work in progress. One of the weakest part is that first singular value decomposition. Right now we're just doing very clumsy scree plot level stuff. I think much more can be done. Maybe with an incorporation of scale space. Also a weakness of what we've currently got is so far when we have more than 2 blocks, Right now, we're just doing the joint overall and the individual. There's things that could certainly be done in between. Right now, we're just working with the reds and the yellows and totally forgetting about the two-way interactions. But it should be possible to do two-way interactions. We just haven't worked out the linear algebra yet, but that's certainly coming. Another interesting area is removal of batch effects. With a lot of these measurements, especially combining data across laboratories, there's big challenges there. One approach to things like gene expression is use so-called spike-ins. What we have in mind is using the spike-ins as one data block, using the gene expression as the other one, and then taking the individual. We have hopes that that'll prove to be a really good way of tackling batch effects. Another big one is supervised JIVE. Notice with the gene expression, I use the subclass information. I only use that information to assess how well they are working. It would be quite interesting to integrate that into the analysis, do an analysis that really uses that information in a strong way. Okay, oops, that got dark. Okay, that's what I've got. Thanks very much.
Questions for Steve while I get the lights back on for us?
Yeah, I have a question related to the signal to noise ratio issue.
Yeah.
So if you have, say, two blocks, and in one block there's like very clear, strong structure, power eigenvalue is like well above the rest. And the other block is more muddy. And you throw this into JIVE,
Yeah.
what will happen?
Yep, okay. Oh, no, I'm not gonna, oh my gosh, okay, let me see. Yeah. Okay, well, that is where a lot of the stuff I sort of shoved under the carpet comes in. Oops. Yeah, okay. Okay, so maybe in fact it's even later, it's a better time. Yeah, this is best. Yeah, okay. Same picture, just different things are highlighted. Okay, so different outputs, you can look only at the patient signatures if you want. And if you do that, you're basically ignoring all the normalization issues. The normalization issues go into the singular values here. And so if you choose to look this way, then you're looking independently of that. But if you want to get into the different types of scaling, then you include that, and that's why that was a separate choice of outcome. Notice all of these, those two are low rank representations. The rank is just the rank of that matrix. Now that can be useful, but the problem is when you do that, you're looking at a rotated version of the data. If you want to come back and do a network analysis, you need to come back with a third one. So that's why these are all possibilities. But basically where the normalization goes away is the fact that it's packed into these singular value matrices.
I'm not worried about normalization. I'm worried, like, let's say the first block there is a real signal, second block there's no signal at all. Like,
yeah.
Clearly it seems like there should be some degradation when you analyze them jointly.
Yeah, if there's no signal, what we hope is we find that at the first singular value decomposition. That's the hope, of course. But so you could even say, well, why do you even bother with the singular value? Somebody asked me this the other day. Why do you even bother with the first singular value decomposition? The reason for that is if you don't, typically, a lot of these data, the rank will be the number of data points that you've got. And so what we're going to end up with is a lot of spurious directions with the principal angle analysis. And so we have to do that to avoid spurious directions at this point. So what I'm hoping is I catch that kind of thing at the first phase. And I agree there's going to be situations it's not. I think it's an area where there's definitely some improvement possible. Yeah.
I'm just trying to understand. So we work sort of on networks combining gene expression and say methylation data using penalized likelihood sort of.
Yeah.
Trying joint and it's not joint. Well, joint and differential network we're sort of looking at. So, but I'm trying to understand. So we're making assumption affects sparsity of the connections, right? Sparsity. So I guess what the assumption you're making here is the low rank of the original matrices, right? You don't quite make assumption of sparseness. Is that correct?
Yeah, that's one of those things I just sort of skipped past. Yeah, that's here. Oops, no. It was in the calculation of the joint. Here, yeah. Okay, so when we do this joint thresholding, we have the problem is the initial singular value decomposition is still art. We don't know how to do that. When you get to the second phase, you got to decide what's joint and individual. There we have quite a precise theory that makes an important assumption of the type you're after, which is we assume that the residuals from the low rank approximation are isotropic. So what that means is you look in any direction, theoretically, of course the data direction is going to be stuff, but look in the distributional direction, then it's the same variation in all directions. So we don't need Gaussianity, we just need the isotropic assumption. I think that's what you meant?
Yeah, well, that's one of the assumptions. I guess I need to understand the algorithm.
Yeah, but we're also, as was pointed out earlier, we're also definitely thinking that our initial low rank decompositions are really capturing noise and really separating that from signal. That's important.
I might just be having a hard time separating rows and columns.
Yeah.
After you've done all these things, to what extent can you make any sort of inferences, such as draw conclusions about individual original variables?
Yeah, that's a great question. Very important research problem that we haven't really solved very well. I did do the kludgy thing with the cancer data of we know the subtypes. And so then I did some inference, but much more direct inference would be great. One area I think we're pretty good at is determining, so there's other stuff I skipped here. We're pretty good at determining the joint. We understand that distributionally pretty well. No. Where was that? There's a nice, here. Yeah. But I had a picture of it. Oh, it's in the cancer data. That's what I'm trying to get here, sorry. Yeah. Here.
The kind of question I have in mind here is, like, would you be able to say this subset of X really has something to do with Y, whereas the rest of X has nothing
Yeah,
to do
there
with
are a
it?
lot of big open problems here. One thing we can say, we have all of our of the space determined by the low-ranked SVD, and then the next thing we do is we look at those and see what's joint. We have a pretty good distributional handle on what's going on with the joint part, and that's what we're actually using here. So I've thought about trying to use the probability distribution here for what could be noise and what could be signal. So we could use that. We haven't done it yet, but that's just one thing I'm thinking about. But you're right, inference is a really central issue, and I show people, like, even show neuroscientists, hey, look, I showed you the picture, hey, we got that right. And they go, well, can you do some inference? Can you tell me how significant that is? I mean, all of these are wide open problems.
Thanks, Steve, on this.
Great. Thank you very much.